Hi, my name is Adnan Ahmed. In this video, I am going to give you a demo about the software that we have made named Wapdap, which elaborates as a wrapper for automated proteomics data analysis pipeline. To use this software through the web user interface, you have to first open the website, then fill out the form and hit the submit button. What the submit button does is it gives you the job ID instantly and also sends two separate email, one to the user and another one to the administrator containing the same job ID. So the user can get the results using this job ID once the process is done, but the administrator has to annotate this job ID with the file path of the raw files for this experiment. To do this, the administrator have to open the path annotation file or script, then copy and paste the job ID in that file and copy and paste the path for the raw files in that file and run the file. Then the administrator have to paste the same job ID into the ID input file, that is an Excel file, and then run the process. From this point, everything is automated. The process first runs the maxquant. In maxquant, first the raw files are loaded and annotated as to which experiment do they belong and how many fractions do they have. After that, the process specifies the parameters to run maxquant, among which the first one is to select the LFQ intensity. What it does is to enable the level-free quantification, and then the minimum LFQ ratio count is given as 1. This represents as the number of peptide to be considered for LFQ intensity calculation. Then it specifies the FASTA file against which the maxquant will search for identification, and then it enables the match between runs option, which increases the number of peptide to be considered for identification. Then the identification and quantification process started. We are not going to run the maxquant as it takes a long time to run. So I'm going to intervene and stop the maxquant. Maxquant generates several result files, among which proteingroups.txt file is needed to run Perseus. I have already placed a protein groups file in place, so the software is able to detect this file and will run the Perseus software shortly and start the statistical analysis process. In Perseus, first the protein groups file is loaded and only the LFQ intensities are taken into consideration for statistical analysis. After loading, the proteins which are potentially contaminant, reverse, and only identified by site are filtered out. After that, a log base 2 transformation on the LFQ intensities is done. This is done to facilitate the calculation of protein expression fold change. After that, an initial histogram is generated showing the distribution of the data. Then those proteins are filtered out which are not identified by at least two replicates in at least one group. Then on the remaining proteins, the missing identification values are imputed from the normal distribution. After the imputation, some data visualization images are produced, which includes a histogram, a volcano plot, a heat map, and a scatter plot. After that, student's t-test is performed to generate the list of statistically significant proteins. Then some additional steps are done to make this data usable for the next result generation step. The result generation step generates simpler results which are easy to understand. This step does basically two jobs. One is to get the protein names from the FASTA file that was specified using the GI numbers, and another one is to sort these statistically significant proteins in 4 different protein groups to be presented. After the process is complete, the software will send an email to the user stating that the job is done and results are available in the website. To get the results from the website, the user has to go to the results tab and copy and paste the ID and hit search. The results will appear as downloadable links. That's the end of the demo. Thank you very much.